My name is Benedict Anchang. I am a Statman tenure track principal investigator at NIHS in the Biostatistics and Computational Biology branch. I head a group on computational and systems biology. Advances in single-cell technologies offers us an opportunity to study developmental processes at both the single-cell and multicellular level. We can now study the spatial and temporal process such as spermatogenesis, which is important for reproductive health using single-cell analysis. Another important single-cell developmental process is epithelial to mesenchymal transition. We can now study such a process using single-cell analysis over time and space. With respect to multicellular systems, a process such as pancreatic development can now be studied using single-cell analysis over space and time. And this raises important questions such as how do we model the expected trajectory of an individual cell or multicellular system. I will now introduce the dynamic spanning forest mixture algorithm, which overcomes the limitations of single tree or continuous models. It accounts for mixtures of discrete and continuous states. It accounts for time cluster and time cluster variability. It accounts for high variable, low variable, skewed, and multimodal significant genes. DSFMix takes as input a time course of staging data and outputs a dynamic spanning forest, which can be directed or undirected. It uses a feature selection step, which comprises of two steps, a shape analysis step, which basically classifies each gene into a multimodal class or skewed class using an FDRA controlled analysis. And it also uses an enrichment analysis to select genes that are cluster specific, time specific, or cluster time interaction specific. The significant genes are then used to build a minimum spanning tree using the spade algorithm, and the dendrogram from the tree is then broken down into forests using a dynamic cutting process. We applied DSFMix to study several developmental processes as highlighted on the slide. For this presentation, I'm going to focus on the process of spermatogenesis, COVID-19 immune response, and epithelial mesenchymal transition. Figure A on the slide shows you the output of DSFMix on a single-cell RNA-seq data of spermatogenesis, comprising of 6 trees. Tree 1, which is a major tree, has a complex two-branch structure where the left branch is associated with the process of spermatogenesis, while the right branch is associated with more somatic cells such as immune cell types. If you look from left to the right of the forest, you will see that the trees on the left are associated with early processes of spermatogenesis, while the trees on the right are associated with late processes of spermatogenesis. Figure B highlights some of the significant dynamic genes that are associated with each of the subtrees in the forest, and Figure C highlights some of the genes that are much more tailored or associated with specific cell types or transitions during spermatogenesis. DSFMix allows us to test for similarities between trees using a multivariate weighted edge-count test statistics. And in the spermatogenesis example, we were able to show that trees 2 and 3 are more similar to the major tree 1 compared to the rest. And all the trees are significantly different to each other. Using DSFMix, we're able to show that gene expression during development exhibits a higher proportion of non-uniform distribution of shapes. And these genes are mostly skewed to the right, and that multimodal genes are enriched the most during development when applied on different data sets. We next applied DSFMix to study the immune response due to coronavirus disease. This disease is caused by the SARS-CoV-2 virus, and it has infected more than 500 million people worldwide and has resulted in about 1 million deaths in the US. It has been shown that 20% of patients who develop severe disease is associated with changes in peripheral immune activity. We applied the algorithm to a single-cell RNA-seq data of about 50,000 cells collected from 7 patients who were hospitalized with COVID. And we also included data of 6 healthy controls. To capture the within and between patient variability during disease progression, we applied DSFMix to the data, and this resulted in the forest shown on the left, where each tree in the forest represents an individual patient disease progression. For example, patient P1 nicely shows a progression of healthy to disease states, and this particular patient was discharged to rehab. And the second forest on the right gives you an overview of the major immune cell types that are implicated in COVID-19 response. DSFMix also allows us to identify key genes that are associated with disease progression. For example, the interferon 1 complex genes show a nice upregulation with respect to disease progression, while the human leukocyte antigen 1 complex genes show a decrease in regulation. To benchmark DSFMix with other temporal or pseudo-trajectory models, we make use of single-cell data from an important biological process called EMT, which is exploited by cells for both normal and disease development. Here on the slide, we highlight 6 visual outputs from 6 different methods that are used for visualizing single-cell analysis. And clearly, you see DSFMix captures the EMT and the MET process nicely. Using the EMT data, we further benchmark DSFMix with other temporal models using similarity network analysis. On the left, we have the EMT reference network showing the transition between the 8 EMT states, and using quadratic assignment procedure statistic, we're able to show that DSFMix and C-street correlates the most with the reference network. During this presentation, I introduced an algorithm called dynamic spanning forest mixture, which can be applied to study normal disease and induced biological processes. It can be used to visualize both single and multicellular systems and correlates well with both biology and real time. I would like to acknowledge all those who played a key role in the work presented, and I would also like to thank ISMB for organizing this conference.